Welcome back. So this is the beginning of the conference in the sense of scientific talks. And it's my pleasure to introduce Sergei Kosakovsky-Pond. He has an interesting career in this field because he is from applied mathematics background, so he can count. But one of the things that he said that sort of influenced how I view data analysis is that he was saying that humans were evolving the ways of looking at things, and just, by looking at things, they can decide whether they're significant or not. So it's very important to plot the data. All statistics doesn't matter. You just need to plot the data. And so when we were trying to figure out who would do keynotes at this conference, we thought that's just the ideal message here. And I'm very glad that Sergei agreed. If you know anything about Sergei, so he made a number of profound contributions to the field of evolution, to molecular evolution, specifically in evolution of viruses. And so he works in this area. So he's an interesting combination of kind of a proper quantitative background with a very complex population and molecular dynamics of viruses. But I don't know if he's going to talk about that today. So thank you.
All right, thank you, Anton. Are you guys able to hear me? I think so. So the idea for this is I would encourage you to download the PDF because it has links to everything else. And I always end up talking too fast, so I generated the Google Notebook LM audio, which just ingests the slides and gives you a 20-minute podcast format, so you can listen to that. It'll be more informative than what I can do. As Anton was saying, I have an interesting, probably irreproducible background because our field is too mature for clowns like me to be successful. But when I was growing up, I wanted to be a physicist. That was back in the 80s. Didn't quite make out. The Soviet Union fell apart. So instead of going to Moscow, I ended up, which is a good thing. I went to a program in cybernetics or computer science at Kiev State University in Ukraine. So Anton is an alumnus as well. Then I moved to the United States and did an applied math or stats PhD in Arizona. The logical progression for that, of course, was to do a postdoc in viral evolution at UC San Diego. And then my first position was in the Department of Medicine. Again, very logical. And currently I'm a professor of biology at Temple. And also, I picked a good time to become an associate dean for research and innovation in our college. The funding environment makes it, let's say, interesting. So the reason I bring this up is that I always find myself in this situation where I can claim to be anyone, really. But what I'm going to try to do today is just give you sort of a little bit of historical perspective and personal experience with scientific software development and data viz. So if I'm known for anything, it would be these things. So in 1998, it really dates me. I guess I don't look that old, but I've been doing this for a long time. As a graduate student, I started working on a program that became basically the workhorse of my career. It's called Hi-Fi. I did not come up with a name. My colleague came up with a name. So hypothesis testing using phylogenies. It's a program that was and is written in C++. In 1998, I'll give you a little bit of historical context. The computational and development environment was entirely different. So at the time, it made sense to develop something that makes no sense to develop today, specifically the domain-specific language. So Hi-Fi is its own scripting language, which I designed and implemented. And it's reasonably used, and it's still going strong, right? So, it's almost 30 years old, and it's still useful. Obviously, it's been upgraded a lot. The motivation for a lot of what I'm going to present in this talk was the web service for Hi-Fi, which I developed as a postdoc called DataMonkey in 2004. And the idea was to allow people that have no experience with software installation, configuration, and don't have computing resources to simply upload their data and run it through the website. So it sounds a lot like Galaxy. So version 1 was written in Perl. I'm not proud of that, but that's what it was at the time. And since version 2, it's all JavaScript, and it's fairly heavily used. And then some random stuff, which is kind of like bring up as anecdotes, because as software developers, if you guys do this, or module developers, you never know what's going to take off. So HIV Trace is a program that's actually, will have a federal contract with the CDC that we developed the software package that they use for molecular tracing. This all grew out of basically what I thought was going to be a throwaway weekend project. that my postdoc then developed into something that is now used by 30 states in public health applications. This is also a web application written in JavaScript using tech like Node.js and D3.js. I'll show you a little bit more later. And then I write modules for data visualization. So phylotree was something that I wrote because it wasn't there at the time to display phylogenetic trees in the browser. They're reasonably well used. Hi-Fi is in Conda, so, 400,000 downloads or whatnot. So to give you a context where I started and where I ended and where I think we're going. So when I started bioinformatics, the sort of like the compute state of the art, I remember my PI spent $10,000 buying this beast called Digital Alpha. It was top of the line, 700 megahertz superscalar RISC chip. For the older people, that might mean something. It was a very hot computer. It ran loudly and it heated the office. And at the time, it could analyze about 10 sequences using sufficiently sophisticated evolutionary model. So state of the art. That wasn't too bad because sequence generation at the time was sort of commensurate with it. State of the art would be something like an ABI Prism Sanger sequencer, which would produce a single 540 base pair read. And I remember quite distinctly when I asked my PI, what is a large sequence alignment? It's going to scratch his head. And it's like, here's this 500 sequences of RBCL, which is a Rubisco protein in plants that's used. It's easy to sequence. It's a good marker. It's evolutionary important. That was a big data set at the time. Computers couldn't handle it. It was a challenge for tree building and such. GenBank, for reference, at the time had only 2 million sequences, which sounds silly. The term bioinformatics didn't really exist, and neither did Wikipedia, so you couldn't even look that up. Python was in version 1, and if you were developing on the Macintosh, you had the old Mac OS 7. How many people in this audience know what that is? Okay, some. It was an interesting system to develop. So Anton reminded me that the last time I came to Cold Spring Harbor was, I think, the first Galaxy meeting that was held here in 2010. And, Anton and I started working on Galaxy-related things around 2007. And that was a typical paper of the time where we demonstrated how to use metagenomic pipelines in Galaxy. I believe that workflow is still available. If you go to the paper and you click on the workflow, the history is still there. I think you can run it. I demonstrated to my undergraduate class in terms of reproducibility and stuff like that. But the point is that, again, 2009, so this was 454 sequencing. At the time, it was big. And, we had big data sets of 80,000 and 45,000 sequences. And the visualization we produced for that was server-side PostScript. So that was a tree that, we generated to display taxon abundance. Now in 2023, which isn't today, but it's close enough, the chip on a previous generation iPhone, which is a quad-core Apple A16 that runs at 3.4 gigahertz, is about 400 to 800 times faster than the Alpha. Modern desktops can analyze easily over 10,000 sequences. Sequencing generation capacity has clearly grown faster than we can analyze it, and it's bio everything. Right? So, bio, every language has a bio module. It's mobile, distributed, and cloud computing abstractions of the operating system. And bioinformatics is such a mature field that the Wikipedia definition hasn't evolved in a couple of years. So it's now an established field. Now I would say, so this is an interesting thing because one of the messages of my talk is that I think whatever we used to do in the past, this is all gone. It's a question of how quickly it's going to go away and what's going to replace it. But there's definitely a feeling, and nobody I think would argue with that, is that in the last year or two, there's this feeling that, this broad concept of AI, large language models everywhere, everything is rapidly accelerating. There's a major, FOMO, fear of missing out, because you might not be doing the right thing at the right time. Sort of a positive take from this. So Andrei Karpathy, who was one of the co-founders, a big name in AI, he was an OpenAI, he gave this keynote just a couple of days ago to the Y Combinator in San Francisco. And one of the interesting points that I pulled out of that, which I think anybody that does development of any kind should take to heart, is that LLMs are a new primary consumer and manipulator of digital information, which adds to graphical user interfaces for humans and APIs for programs. So whatever you do, especially if you start new projects, build with agents and LLMs in mind. So now I'll actually go back and give you some specific examples of just cartoons of what I do, show you some visualizations and tell you, what we have been doing and possibly expose you to some new ideas that you might have seen, might not have seen. So Biological data means many things to different people. That's one. That's the type of biological data that our tools consume. So HiFi, it's a sequence alignment, which is what's displayed here. It's grouped into codons, which implies that it's coding sequences, and they're color-coded by different codons. So each column, if you're a statistician and you want to study the evolution of this, you have an evolutionary model that tells you something about how this evolved. In reality, what really matters for evolutionary process, and this kind of points to old-style data visualization. I'm sure you've seen this before if you looked at multiple sequence alignments, that it doesn't matter what's there per se, it matters what changes. So if you basically abstract everything that's constant and only look at the variation, you will see such, a reduced plot of the alignment. These sequences are all related by descent. So the right formalism is not to look at them as tables of character data, but rather look at them as evolutionary trees. So that's an evolutionary tree. I'm sure everybody has seen something like that at some point. So you have existing sequences at the tips. Internal nodes in the tree represent unobservable ancestral states. There's some evolutionary divergence or distance associated with that tree. Sequences are related in evolutionary tree, and we'll have a statistical process that models evolution along each branch. So an example of, say, what a comparative analysis and, say, HiFi and DataMonkey would comprise, and a lot of people do this. Again, I don't expect many in this audience have done this specific analysis, but it doesn't really matter. It kind of all follows the same workflow. So you obtain homologous coding sequences through a database search or some other tool. It's external. You align them, preserving reading frames, which is a bit of an open research question, how to do it properly in large scale using other tools. Then you infer a phylogenetic tree, also using other tools. You select suitable analysis options through command line interface or web form, and then you run a specific type of analysis called DNDS-based analysis to quantify the extent of natural selection acting in these sequences. So this can be done server-side backend or in the browser. This returns a detailed JSON, so I'm highlighting the procedural implementation, structured, human-readable, machine-readable data set, and a markdown summary that you can read. And then the way we present this to the user, and this is what Anton was indicating in terms of exploratory analysis, is using an interactive JavaScript notebook using this particular platform called Observable. So, you can load it up on your phone or device if you want, or you can just follow along. I'm going to show you what these notebooks are like and kind of illustrate some of the concepts along the way. And that'll tell you why we're doing it this way. So, just for the programmatically inclined, everything is done fully in the browser. I'm not hosting anything. I'm hosting it on this website that's free, observablehq.com, that hosts. It doesn't have to be that. You can host on your own systems, but it's, it's host free, compute free. Everything's done on client side. It pulls down a JSON file and renders it as so. If you're a biologist, what I would like to sort of illustrate to you here is that there are different degrees of understanding of what analysis tells you, right? You've all run a linear regression, right? The primary outcome of linear regression is, what do you think? Well, what do people report for linear regression? What is the one thing that you see? R squared, okay? So here it's a p-value. Would you actually be comfortable doing just that? Running a linear regression, reporting an R squared, and saying you're done? Probably not. You should at least plot the data. You should do some assumption testing, whatever. So you should visualize these types of data. And for linear regression, it's easy to do because you have 2 dimensions, scatter plot. You directly process it. This is a result which tells you that, based on the likelihood ratio test, there is evidence of episodic diversifying selection in this data set. Great. You can take it there, or you can start looking to see, where this signal comes from. And, for example, here, when I mean reactive notebooks, for example, I can change, the stringency level, and everything will update automatically. Again, in the browser, no server communication. So for example, and these are specific plots that we've generated to visualize these data. So here, it, for example, tells you higher, values indicate that this particular location in the alignment has some evidence that there's selection operating in this alignment. So what this tells you immediately that selection, if it's operating in this alignment, is non-uniform. There are hot spots and cold spots. You can look at it in even greater detail, for instance, by looking at not just sequences, but sort of individual matrix, right? This is a heat map that tells you, there's this specific site 48, that happens in an internal node. There's a particular substitution that has a very strong statistical support for positive selection. Okay? You can look even further. You can say, okay, so what is the, what is the site that gives me the highest evidence of positive selection? Codon 110. Okay, and now it's going to display to you the phylogenetic tree at that site. Again, everything in the browser. So you can see now which branches in this tree corresponding to which characters give you evidence of selection. Like if you're an evolutionary biologist, if you think natural selection, if you know anything about amino acids in the protein, there has to be some change. And there probably has to be more than one change. And if you look at this tree, the strongest evidence of selection happens in this branch, where there's a substitution from valine to a cysteine. Right? And you can also see that this, for example, this involves, these types of codons. You can see the individual alignments as well. But the point that I want to say, is everything is draggable, quickly updatable. So the main point here, from the standpoint of development, is that this, I can guarantee you that this is going to work, unless the internet goes down. Because, all I need is an internet connection and a browser. I don't need any dependencies. I don't need installation. This is going to work, right? And it's going to work on your phone. If you can try it, perhaps it's not going to render as well. It's going to work on any device. So this is a big plus for me. So in 2021, there was this little opinion piece that came out in Nature, Reactive, Reproducible, Collaborative Computational Notebooks Evolve. So I'm cited in it, but it's actually an interesting piece. You should read it. Historically speaking, I would think that the first package that created what is the modern notebook is Mathematica. I used it when I was a graduate student in applied math in 1998, and I was blown away at the time. It was pretty slow, but it was doing amazing things. So Stephen Wolfram, who's the developer of Mathematica and a mad genius, for many reasons, I was impressed with how it could compute analytically integrals in 1998 using cellular automata. Now things are more impressive. He called it computational essay. So I like that. So they're now about 30 years old. I'm sure everybody in this room used some version of notebooks. So Mathematica was introduced in the 1990s, and then IPython and Jupyter in about 2010. They're very popular. They're about 10 million in the public domain. And I think what notebooks recognize is that much of the scientific data visualization is non-standard, meaning that there's no one way to visualize the data until, with very few exceptions. But that is also built from many standard pieces with some degree of customization. So you don't want to run from scratch, but you also don't want to have just a limited number of templates that you can select from. For modern data presentation, I think an important factor, and you can see that it's important because everybody's starting to adopt it, is reactivity, which means that notebooks automatically update when input changes. For example, traditional IPython and Jupyter notebooks do not do that. Newer versions do in some sense. If you want to, since you're at a Galaxy conference, you should care about things like versioning and collaboration and pinning specific packages. You can do that. There are very flexible ways of ingesting primary and pre-processed data. And what I would say, and this is going to be a refrain, it becoming less and less relevant what it is that you use, because I think it's going to be much, much easier to move between systems. So the choice of the actual notebook system is not important. I'm presenting observable because I work with it efficiently, and I like the guy who designed it, but it doesn't have to be done that way. So observable uses a reactive model of code execution. What that means is that it knows how different cells depend on one another in the notebook. When any cell is updated, all cells that depend on it are automatically rerun. And this works with any number of cells, and their order in the page doesn't matter. So when I show you the next notebook, I'll show you what's actually in the cells, self-documenting as well. So here is a table for your reference. Don't expect you to read it, obviously, of some of the more common notebook systems. So, you got Jupyter, you got RStudio, which is now Posit, Google Colab, Kaggle notebooks, DeepNote, and Observable. You notice that most of them are fairly recent. So the oldest here is 2011. They're all widely used. The one thing that's interesting is that with the exception of observable, you see all the traditional ones say no, but then there's but. So the original implementation didn't have reactivity, but now you can do it. So people are appreciating that this is increasingly important. You know, and some of them are free, some of them are not. And this is not a complete list. So this is a very, very busy space. There's commercial offerings and it's not settled. But, you know, there are lots and lots of different options. I'm sure most people in this audience would, you know, line 1 and 2. Sorry, the other thing that I want to point out is that observable is the only one that's focused on JavaScript. Most of the other things will have traditional data analysis languages, as things like Python, R, Julia. What that tells you to do, I mean, from a technological perspective, this forces you to basically run non-native code from the standpoint of the browser. So you invoke a kernel, you connect to an external machine, you can run an emulation level, so you can actually run Python within the browser through Pyodide and things like that. But there is a layer of indirection. It's like you basically virtualize your language within the browser. JavaScript, on the other hand, is native. So it's the de facto language of the web. Recent estimates, I don't know how you get it, but you know, I haven't verified it, but I believe it. Over 98% of sites use it. It's natively supported on all modern browsers, on all modern devices, and it permits in the browser execution. I'll talk about it a little, actually quite a bit more later. Is remarkably fast and getting faster. By many measures, it's the most popular development language, believe it or not. It has a vibrant and large ecosystem. It has increasing robust backend support, which means you can run it server-side as well, and it has a rapid development cycle. So this is an interesting history of JavaScript because, again, for your reference, but originally JavaScript was designed by this gentleman called Brandon Eich for Netscape. Probably some people remember what that was. Then for a couple of years, there was, you know, Microsoft did its thing. And again, for a lot of you, this was ancient history, but Microsoft did what Microsoft always does, which it reverse engineered JavaScript, created its own incompatible version to try to get people to use Internet Explorer. You know, in 1997, 1999, this was all standardized. So JavaScript became a standards-based language. Then the next sort of waypoint, which we all use this every day. So you might not be aware of this tag, but it's all there. So Ajax, which is an asynchronous data fetching, was 2000, 2005. 2006, jQuery library was released, which is a way to manipulate elements on a web page. The next big thing was 2009, where Node.js is a package that was developed by Ryan Dole to allow server-side execution. And then the most important thing that you see now is that a lot of the modern web development and interface development is based on what's called modern frameworks that came from commercial entities. So there are JavaScript frameworks for rapidly developing interfaces and visualization abstraction. So React came from Facebook. Angular was eventually done you know, by Google, then Vue.js, and so on. And this brings the concept of single-page apps, which is something that was very foreign in the 1990s when I was starting out. So multiple windows, multiple pages. Now it's a different paradigm. So JavaScript is really fast, which is surprisingly, you might not appreciate it. There's a school, again, if you care about these things. So Debian, which is a Linux distro team, they have this constantly updated benchmark, with C, the compiled language, setting the reference. So it's 1, and then you see, C++, Rust, C#, blah, 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 Java. So Node.js, which is a JavaScript indication, is only about 3 times slower than compiled C. That is insanely good, and it's getting better. For reference, here's Python. It's 100 times slower, unless you do foreign libraries calling. R is not in any of the benchmarks. I try to find one, but nobody does it in this space. So if somebody wants to do it, if somebody wants to implement the basic like in-body algorithms in R, go for it, but I don't think you want to do it. But it's slower than Python. If you do, unless you do external calls. So JavaScript is very popular. So this is a recent ranking of what developers think are languages, one, worth learning. That's the first column. So Python, C, C++, Java, C#, and JavaScript. Stack Overflow again, this is more like what is important to know for you to develop. So JavaScript, HTML, Python, SQL, TypeScript, which is a typed version of JavaScript, and Bash. Many people program in Bash. Some, but it's there. Good for you. And then GitHub Octaverse is sort of a sense of how much code base there is for it. So Python, JavaScript, TypeScript, Java, C++, C#. Again, any of the scientific packages would be like off the page. This is general development. And it shows you the trends. So this is a beautiful trend that that GitHub generated. So, and you can see, who can tell me what made Python the most popular thing in 2024? AI, yeah, that's it. So everybody's using AI integration, but then in everything else is still there. But what I would say, going back to what Karpathy was saying, the language I don't think matters that much anymore, since a lot of low-level development is or will soon take place in human languages. So, I mean, we may not be quite ready for it, but I think we're to the point where, if you're writing in script with, or programming language for some applications, it'd be like trying to write an assembly in the 80s and 90s instead of using a compiled or interpreted language. So I think a lot of programming is about to take place in human languages. So I mentioned the JavaScript ecosystem is huge. The main package repository is called npm.js, Node Package Manager. It has over 3 million packages. It is the largest software registry in the world. If you care about visualization, the modern way to do visualization is some sort of a GG, grammar graphics package. So there's some that are exist in the JavaScript space: D3, Vega Lite, Plotly or Observable Plot, not an exclusive list. Here's a lot of bioinformatics packages that exist in JS. Do you know? Do people know any of them that are in JS? And again, I didn't put the years on it, but this is all recent development and it's accelerating. So I think this is a trend that you will see. So, you got IGV, JBrowse, Site-Escape, etc. So let me show you a few other examples, fairly quickly, just to demonstrate that you don't have to. So this was an example from COVID era, which we're, trying to quickly put out results of complex analyses. So for example, here, there's a Vega Lite specification of this graph, right, which is directly in the page. So what this, just to give you a backstory, we're running genomic analyses of large collections of SARS-CoV-2 genomes and trying to annotate things as they were changing. So here's a cartoon of the SARS-CoV-2 genome. Here's some sites which are interesting. You can look at a specific site, look at its what its selection profile is over time, frequency trends, so which residues exist at a particular time point, and get replaced by others. So this was N501Y, the first named variants, how are they distributed across different countries, and how they're distributed on the tree, right? So the real, so take-home message from this is this didn't take me long to develop. I mean, everything I felt at the time for COVID, getting things out quickly was important. So I think this was about, I don't know, a day and a half of work to get this all done, and it's still there and it's still live. And then another one that is now directly related to Galaxy, where we're basically using Galaxy Analytics pipelines for COVID to cull variants from deep sequencing data sets. So this represents sampling frequency over time in this data set. So this is a result of an analysis of 94,000 deep sequencing data sets. And the main point was to look at interhost variation. So do you have within host evolution? So here you have a plot of, what type of interhost variants do we see, indel, synonymous, non-synonymous, and so on, frequency of a particular variant. But again, you can, if you're interested, you can look around just to show you that these are all, this goes back to what I was saying, that each visualization is more or less custom made, and it's not directly transferable to a different application, but we can use the same building blocks. So notebook integration is observable. Notebooks run in the browser can retrieve result files in many formats. Again, it's true of all modern notebooks, so you can do JSON, you can do text, you can do SQLite, you can do database interfaces, you can pull things from S3. Observable and some other systems can be embedded, so you can actually put them within a bigger application if you want, and they all provide data export and import. So next thing I want to talk about is the benefit of moving some compute to the browser. And what I mean by this, and I'll come back to this in a second, is that the traditional, and everybody does it to an extent, right? You do some validation in the browser before the data leave it. But what I want to emphasize is that it's becoming progressively easier to do more and more. And why is it beneficial? I mean, I'll just give you a couple of obvious bullet points. So it's enhanced privacy and security, because if you do client-side processing, the data never leave the browser. It achieves near native speeds for complex calculations, and you can process remarkably large data sets directly within the browser. It obviously reduces your cost for infra. You don't have to run servers. You don't have to deal with fragmented traffic requests. So offloads computational workloads from servers to end users' machine. It's real-time interactive analysis. Again, there's no delay because you don't have to communicate with the server. And it actually allows for code reusability if you do it properly, allow the use of established and powerful data analysis libraries in Python, C++ directly in the web environment. So the technology that grew out of this particular effort to do native time or native speed client-side compute is WASM, which is WebAssembly. And it started out as basically being a subset of JavaScript, which made it easier to do just-in-time optimizations. So they basically reduced the language feature set, but now it's much more broad than that. It's a low-level binary format, which is all implemented in the LLVM framework, bytecode for near native speed in web browsers, significantly faster than JavaScript for computationally intensive tasks. The exciting thing for me is that it's no language agnostic. You can actually take code that's written in standard languages, transpile them to WASM, and run them on the web. I'll give you an example of what we did for that. You have a secure sandbox. It executes within a safe environment in the browser with no direct access to the underlying operating system. It's designed to work alongside JavaScript, allowing developers to optimize performance-critical features while using JavaScript for UI and other tasks. And it's portable and open standard. It runs in all modern browsers and can be used outside the web, making it a versatile and future-proof tech. So in fact, there's a new thing called WASI. So WASM you can think of this as WebAssembly to run in the browser. There's something called WASI, which is being very heavily pushed by big players in cloud space, because this is a possible technology for virtualization, where everything is written in one language and it runs server-side, client-side, and clean. So they do, for example, WASM doesn't support multi-threading and some other features that we care about for high-performance compute, but WASI does. So watch this space. Things are happening. Everything is standardized and options are actually getting reduced. So use case of less than 1 day of work, where I wanted to see, it was about last year, so the tech is a little better now, to do complex evolutionary analysis directly in the browser with HiFi. HiFi is a big C++ package. So I wanted to see how long it would take me to, port it in the browser calculation. So I used the mscript and open source toolchain, which basically takes your standard C++ or other code bases and compiles it to WASM. It was more than compilation because I used some weird things in C++ that weren't working in JavaScript, so I had to do a little bit of fixing. So what this does, it creates, a WASM binary with data that you can upload to the server. There's an extra layer that you have to do for standard application porting because WASM doesn't have an idea of file system and user UI. So you have to wrap, if you want to read from files and write from files, which is what most bioinformatics tools do, you have to have another thing on top of it that mimics it within the browser. So there's this other thing called BioWASM, which is a product that I think originally came out of the Chan Zuckerberg Institute. I think the guy that started it is now has his own company. But BioWASM.js, look it up. They have really cool in the web demos for standard tools. They provide a wrapper for file system operation. So then I modified HiFi code to provide real-time updates to the browser. It took about 2 hours of effort, and I pushed it all within an observable notebook. So here's a notebook that takes my 100,000 lines of code binary that's been compiled into WASM. So it's now loaded. It sits in the browser. I can choose an alignment, from a file system. So I'm going to do, I'm just going to run it. So I'm doing an evolutionary analysis straight in the browser. So it actually gives me updates as it's doing it. So it's emulating the terminal. And I want to emphasize that, my benefit. So what I tend to do as a developer is do the minimum amount of work that I don't want to do. And that, I think, is a good indication of how you can basically build something that looks reasonably polished and executes things in the browser with minimum amount of effort. And I didn't even start. So I stole this notebook. So you see, this is a fork of Hello BioWASM, which some other user at Unobservable produced a template for how to integrate BioWASM into notebooks. So I just forked it, edited it. And, so the analysis is progressing. I mean, to be faster at native speed, but it's still doing reasonably well. Have real updates. And again, this is all running in my browser, right? No server side, and I can click on the results, and it's going to take me to a dedicated page that shows all it is. Okay, so some other WASM applications in this space, and this is sort of not quite there, but you can see it's starting to take up, just to give you an idea. So there's BioWASM, which is a collection of common bioinformatics tools compiled to WASM. You should check it out if you're curious, because they have, they did things like, they have FastQ, and they have aligners, and they have some basic sort of standard tools packaged. You have FastQ Bio, which is an in-the-browser calculation for, FastQ analysis. You have Viral WASM, a suite of tools for viral genomics. And you have some other stuff like docking, molecular docking, or you have an application for interactive single-cell omics data analysis. Since you guys were talking about it yesterday, I thought I'd put it in. Also fully within the browser. This is a little fancy. This is not quite polished because you have to run servers in your system. But anyway, so why, so to summarize, everything up to this point, I think I'm doing all right on time. I want to leave some time for questions. So I've, the browser-based notebooks for visualization, to me, are sort of, by and large, what should be done for most, say, exploratory development, anything short of like high-end repeatable production. So rapid prototyping, very important. Instantly code, viz, and develop. Universal access. So it's important here. You don't need specialized software. You have a web browser. Everybody has that. Because you have rich interactivity like zooming and filtering, allows you to play with the data and discover patterns, which we're very good at. We have a lot of brain cells dedicated to that, even though I was shocked to find out how low our visual bandwidth is. Does anybody know the ballpark of the visual bandwidth that the human neural system supports for passing, visual information down to the brain? Order of magnitude. Good guess. Yeah, it's 1 megabit, a little less, I think. It's pretty impressive compression if you think about it. But yeah, so we're like low rate people. Low rate systems, but we do that well. And then effortless sharing. One thing, I can tell you if all I need is a browser, for demo visualization, I don't need anything. I don't need servers. I can even do it without an internet connection. And I'm going to conclude by basically telling you where I think everything else is going. And I'm doing it from a perspective of somebody with a relatively long career. And everything, and quote, I've done or want to do seems already obsolete. Because of quote unquote AI, whatever that means. And I'll give you a couple of examples. So, and then as far as like the minuses for that is that when you build a scientific career or develop your skill set, you're invariably, to be competitive and productive, you build some sort of a moat. You develop skills that are hard to replicate. You develop tools and maintain tools that solve a specific technically challenging problem will be difficult to replicate. You do something to make yourself unique. And I think there's definitely feels like this might be disappearing, which is a challenge, obviously. You know, it's like a severe selective bottleneck for everybody, perhaps. The tech is evolving very rapidly. I mean, the things that I can show you today, or at least talk about today, weren't possible 2 years ago, right? And I'll give you one example to show you how quickly this develops. It feels like it's hard to keep up. You know, on the positive side, you can automate boring tasks, such as stock UI UX design or most scripting, some optimization and code refactoring. I think, you know, I guess embarrassed and proud to say that, you know, I'm to the point that I think vibe coding is something that we should try as, you know, interface and in our field for rapid prototyping and design. There's a lot of interesting developments in LLM AI-based interpretive data viz. And then where you've seen a lot of applications so far, which I think is sort of misusing the tools, is result interpretation and discussion, right? So somebody wraps a chatbot around a paper and says, talk to me. It's like, I don't find that as a compelling application until you do something, you know, more interesting. People don't like to stare at a prompt. You know, you need to, some people do, I don't. So the use case, all right, for DataMonkey, you know, what I mean by vibe coding, and again, going back to this, is history. So our lab has been developing DataMonkey, which is a one-button comparative evolutionary analysis platform. That was a lucky design decision for me at the time. It made the platform very popular. 75% of our users and citations come from the web platform. This is a screenshot of the 2004 interface, which was based in the traditional Perl and CGI architecture using SQLite per session storage. Everything was done server-side. There was no framework for UI or UX. I designed that, you know, so I can take blame for this, for these visual cues, although I do like the color scheme. So this was all hand-coded. It was implemented as a collection of Perl, Hi-Fi batch language, and R scripts using GNUplot, GraphViz, and GoScript for visualization. So that took me about 3 to 4 months to develop. You know, and then some refinement, and it was that's published in 2005. Then version 2, which sort of came about 10 years ago, so it was published in 2010. So this was all JavaScript. So Node.js web application with MongoDB storage and cluster execution. We used one of the frameworks that I mentioned for UX design, React. We used D3.js from the browser visualization, moved some of that to observable. Apart from the viz, everything else is done server-side. And that was written by a team, including a UI UX designer, and took about a year total effort, on and off. One issue with a system like this, which kind of points to, I'll come back to this, is that once a system becomes complex enough, and this isn't complex enough, but it's just not too complex, but it's complex enough, nobody knows expert knowledge. No single person has expert knowledge of the entire tech stack. So updating and upgrading becomes slow and untenable, unless you do something else, like Galaxy has a community, you know, better documentation. So that is a big problem, you know, especially when developers leave. And most of the DataMonkey analyses are also in Galaxy, so you can, you know, run them. And version 3 was something that we're just playing with this year, is a prototype single-page application. We want to get away from React because it's a system that's difficult to maintain. I don't like it. So there are better JavaScript systems. I don't want to learn them. I don't want to care. I do not want to learn about another UI UX design system. So, you know, I asked my developer, Stephen Weaver, who's not here, but he's sort of been instrumental in old DataMonkey designs, is to sort of start with these core principles designed to provide researchers with an intuitive interface to validate, analyze, and visualize sequence data without requiring server-side computation, to bring analyses methods directly to your browser, and the workflow built around clarity and efficiency. And the core principles is intuitive workflow, progressive disclosure, browser first, and local privacy. And this is his developer workflow, right? So I asked him to show me what he's actually doing, is, you know, collaborate with Claude Code and MCP browser to identify critical UX issues. So if he has an instance of Claude interacting with the browser, interacting with the site, identifying issues that it thinks need to be replaced or improved, you know, after a while, outline streamlined solutions to quote unquote developers, then pass that to another instance of Claude that implements those with some human supervision, and then you iterate. So you can leave it alone. And Stephen was telling me that Claude at some point had a, what was it, a risky commit. So don't ask for anything, just do everything on your own. But I think Anthropic disabled it because at some point Claude hacked the system. So it is now considered dangerous. Not in our application, but you can basically have a completely independent iteration. So ensure all development is done in feature branches as things go wrong quickly, and developer becomes more of a project manager. So, show you what you can do. And this is a prototype of developer time so far. Oh, come on. Thank you, Bitly. It failed. Congratulations to me. Okay, I'm going to go to Bitly and look up the direct link. There we go. So yeah, this is, so this is a Claude design system, which allows you to basically upload files into your system, perform validation, view trees. Do analysis, select a method, and it goes on and runs it. So it does things that I don't care about doing. I don't want to learn Svelte. I don't want to know how to store data inside the browser, which all it does. I don't care about persistent session storage. It's all a nuisance. I don't care about how these visual cues are designed. I just want to have things look a particular way. So, and the fact that you can do this with basically minimum, you start with a design, you guide Claude towards, what you want it to do, you interact with it, and it basically builds something that, I don't know, if I were starting to do this from scratch, that would take me, a couple of months just to get to the point where I understand Svelte to get it done. Now, the important thing about this is that this is not what I would call mission critical, because, there are a couple of things here. Visualization is easy to test, right? You just know if it's right or wrong, and you can do test suites. And the proper design for all of this, you need to have a test scaffold that confirms that everything that you do is correct. But you wouldn't necessarily do it for statistical analyses where you care about the correctness of the results without additional checking. You can do it, but it's more work than that. So, and then there's this other domain, which, and the history here is fascinating. So it's natural language to NL to Viz. And this predates large language models. It's this idea of using, some sort of machine learning to transform natural language to visualization. So the seminal papers, the first seminal paper that kind of started this was in 2007. And then until 2023, basically what they were doing domain-specific representation, building custom models, representing, converting language into tokens, specialized tokens, mapping these tokens to specialized visual cues. And now you can see basically everything in 2023 and from that point on is large language model. And here's some applications of natural language to Viz for loosely speaking genomic or general space. So there's OR genome, circular genome visualizations, there's biochatter, there's general scientific visualization, including one from Microsoft. But if you've never done this, I would encourage you. Dannon was showing Claude MCP yesterday, which is a much more sandbox and guided interaction with a particular system. But I would encourage you to, take a result file from any of your software packages that not too big, something like JSON and CSV, give it a little bit of context and ask you to interpret it. Any of the major chatbots, you'll be surprised at how good they do. All right, in summary, so what I attempted to show you was a little bit, high level. That genomic analyses benefit from some in-browser computation using WebAssembly, allowing many tools and steps to run directly on a user's machine for better privacy and lower service costs. Reactive computational notebooks enable real-time exploration of complex genomic data sets. AI and LLM accelerate research by automating coding tasks and potentially generating viz from natural language commands. And the integration of these tools is close to enabling a single developer workflow I don't think we're quite there yet. I mean, you have to be careful about how you do it, but we're getting close. Where AI is used to build browser-first applications and enabling rapid prototyping and universal access to interactive analyses. But I think, the way I tend to view this is that, if you step back from, in my personal experience, any coding task for implementing and developing a tool, it's the standard 80-20 rule, which applies everywhere. In this case, it means you do 80% because you must, and you do 20% because you want, right? So, you have to do all these things. It's not interesting, package management, build scripts, distribution, some things that just have to be done in order to do a proper package development or, shell scripts and stuff like that. So I think the easiest thing, and I'm really finding it for me, I can already see this happening in our workflows, is that you can reduce the 80% to probably more like 20%. So now you can offload most of the stuff that you don't want to do to automated agents. All right, thank you guys. And then I'll put up the, here, let me put up the first slide. So if you want to just, screenshot that QR code and you can get the PDF. Thank you.
We have 5 minutes for questions. I'll try to run as fast as I can. Oh, okay.
Thank you so much. Fantastic presentation. I was wondering, we get so much benefit from this dynamic visualization. Do you think, like in publication, we're going to be limited in the future by this flat PDF format where we have to like one static visualization? And do you think this kind of like collab notebook can be used like as a integrating dynamic visualization in a PDF paper or something?
It's a great question. So, obviously, I don't know what's going to happen. It does feel very much like the traditional mode of scientific publishing and distribution, which is a static document, is no longer meaningful. It's still there because there's inertia, but you want to have something that is interactive. You want something that is collaborative and updatable. So there is some effort. I personally, what I've tried to do in the last couple of years when I do scientific papers, I publish GitHub repos with observable notebooks for people to replicate things, mostly because I don't want them to experience the same frustration that I do when I try to find, what other people have done. Now, whether or not the system will move in that direction, it's anybody's guess. But I think looking in the future, that would very much be a desirable direction. Have interactive, live, what did Wolfram say? Scientific essay.
Hey, hi. Thank you. This was a really interesting presentation. My question surrounds the use of JavaScript.
Okay.
Particularly, JavaScript ecosystem can sometimes, like there's parts of it that are extremely brittle, like the left pad incident that happened a few years ago where one package broke in NPM and then there was a cascading failure. How do you think this sort of work will hold up against that?
It's a great question. I don't think that, so the fragility of any large enough software ecosystem, it's not special to JavaScript. It's probably just exposed in JavaScript because if it goes down, everything goes down. You can see just in the JavaScript ecosystem, the evolution of package managers and distribution systems in the last couple of years. It is not perfect, but there's been so much effort put into it that I think we're going to get to a manageable solution. But all of these, package management becomes, in some sense, a problem for any language system. Python package management, virtual environments, R is not as complicated because, it's simply, a more circumscribed language, but you can have similar issues there.
And it just broke a lot of packages in Bioconductor.
Yeah. So, the standard solution to this, I think, would be to, if you're really diligent about this, you do pinned versions. This is so to make sure that you don't just include the latest version of a package, but you do, specific set of packages. in that case, it guards against that in some way. And I mean, Galaxy, I think, Galaxy encourages you to do this as well, because your workflow is not just packages, but specific versions of these packages. It's not ideal. It's a lot of hassle. I agree with you.
One last question.
Thank you for the presentation. I would like to ask you that how much data you can process on the client side?
Well, I mean, all right. So there are a couple of limitations to keep in mind. So the biggest limitation is that most of the browsers will put a gigabyte RAM limit of what can run within the browser. So if you have something that is very memory hungry, you will have a problem. The other thing is you have to be careful about how long it takes to run anything in the web framework, because people, the sort of the entire design paradigm is that things remain responsive, right? So if you go into the browser and it just hangs there and does something for 30 seconds, that is, not acceptable to the user. There are obvious ways to do it. You just shuttle off in the background process. But I would say, you would, what I view this for now, for our applications, is you do all of the data validation client side. We can do it for us. Some others, like if you're uploading images or single cell data, that might not be viable. And then you offload high compute, anything that takes, say, more than 5 minutes of compute time, you send off to the server. But there's a surprising number of small tasks that you can do that require less time than that. And I think it's just going to, it's surprisingly fast. I mean, you can run the analyses in your iPhone. Like I said, it's 800 times faster than that $10,000 digital alpha. But obviously, for genome assembly, you're not going to do it.
Hi, so really cool talk. Really enjoyed the talk and showing the power of what you can do in like a browser sophisticated computing. So I'm just kind of wondering if you could comment a little bit about like concerns about code provenance and kind of ensuring. So just to give some background, so like in the JavaScript world, there's this idea of minifying your JavaScript, right? You convert all your variables into these single, letter representations, remove the spaces, remove the new lines so that it can download efficiently. And now you're going, further in that direction into having binaries. So how do we ensure like that? You talked about pinning versions, but how do we ensure that, all those software are available to people, just giving an application, in a web browser, but you can go back and make changes to that step of how that was developed and, sort of modify that web application to be more, to make variations of that?
Well, I mean, so there's always, I think, if I understand your question correctly, is, you might lose the connection, between the binary that you download and the original source code. I think that's true of any system, right? To a degree. Any JavaScript or any other package that you compile to WASM, at some level it exists as a source code tree somewhere. And good packages will give you, the vast majority of packages that I've come across, they give you the whole, thing. There's a GitHub repo with the underlying code, there's a build script, and eventually what gets delivered is not that different from the executable binary in Linux. But code provenance, it's not something that I think is being considered in the way we would in the scientific computing space, because there it's, it's more of a commodity thing. It's like you don't really care, who did it so long as it. But I haven't thought about it in any detail. So I think there's a potential concern there, but but I also think it's the same for other distribution mechanisms, don't you think? If you download a binary, right? If you download a Docker container that has no source code.
Yeah, so I think the efforts are, I don't know if you're familiar with SPDX identifier. So every, source code file has like a machine readable identifier of what the software licenses, and then you have a software bill of materials. And then it would just be nice, I think, if these types of like products come with something similar, or there's a capability for that,
Okay.
right? So,
it might very well exist. I'm just probably not aware of it.
okay.
Well, let's thank Sergei, and he's not going anywhere. And I think we have a coffee break now, right? So You can talk to him at that. Thank you very much.
Hey, so good morning. My name is Sehyun-Oh. I'm an assistant professor at CUNY, and today I'm gonna moderate this for our first oral session. So before we start, just a quick logistic thing, mostly for the presenter, but for the audience at the same time. So we have 9 presentation for next 1.5 hour, very tight schedule. So each presenter have 10 minutes, and then once its timer start, the green light will show up, and then 8 minutes point, like yellow light will light up, and then I want the presenter kind of wrap up your presentation within a minute or so, and then we will have a 1 minute of Q&A session. And also if you have a question, just wait until you get the mic, because so the online people can hear your question as well. So we need to move on quickly. So for our first, so this session is covering a lot of covering mostly the Bioconductor and the Galaxy's infrastructure. And then the first talk is from Junhao Chu from Moffitt Cancer Center, talking about Galaxy all-in-one agent, unified natural language interfaces for cross-galaxy data analysis.
Okay. Good morning, everyone. I'm Junhao, a software engineer at Moffitt Cancer Center. Today, I'm very excited to introduce Gaia, Galaxy all-in-one agents. Gaia leverages large language models, LLMs, to provides a single chat interface that unifies all your registered Galaxy instance and recommend tools, run tools on your data sets, and manage data sets across multiple Galaxy instances, all from natural language commands. And the first question would be, why Gaia? We identify 3 core challenges that some Galaxy users might face. First, Galaxy instance can have overlap, but different tool sets, making it hard to determine which Galaxy instance to use for specific data sets or tasks. And this also leads to the second challenges. A lot of times it's lost just hunting for the right tools. And the third challenge is that sometimes if you want to run the tools, but the data sets are often scattered across different instances or histories. And Gaia provides a solution for each of these. For different tool sets, it offers a unified chat interface that can communicate with all your registered Galaxy instance. And to reduce the time spent tool huntings, it provides a smart cross instance tool recommendation and executions. And it also provides the automated data discovery movements. And the first time new users see this page from galaxyproject.org, they might wonder what the difference is between these different server options. And not just for new users, even for experienced users, they don't always know how tool sets differ across different servers. For example, experienced users, Galaxy users, data scientists, and Moffitt has asked me exactly the same questions. And yes, we have a lot of training tutorials, training events like Galaxy Training Academics. They're very helpful in this case, but this is also something Gaia can help. Instead of manually checking each server, just simply ask the Gaia's to recommend a tool for your data sets and tests. And then Gaia will search the tool across different Galaxy instances and pick the most relevant tool for your test or data sets. And sometimes it's just hard to run Galaxy tools, when the data is spread out. And imagine that you want to run the Galaxy tools on use Galaxy.org, and then you already know you need 3 data sets, and you already know that data set 1 is on use Galaxy.org, but you don't know where data set 2 is. It might be on another Galaxy histories, or it might be on another Galaxy instance. And you have data set 3 is already selected on in Gaia. And then you ask the Gaia to run the tool, Galaxy tool for you. And the Gaia will first search for the history containing dataset 1. And then it will search the dataset 2 across different Galaxy histories or different Galaxy instance. And Gaia will move dataset 2 and 3 into the Galaxy history containing dataset 1. And with these all 3 datasets, Gaia now can figure out how to run the tool you want to run, and then run the tool for you, and return the complete schedule back to the user. And then you can go directly to the Galaxy history to view the results without the need of worrying any kind of intermediate steps. And this slide shows how Gaia can select and execute Galaxy tool from start to finish. Here, a user asks for a machine learning classification test on the selected data sets, and then Gaia takes the request, identifies the most suitable Galaxy instance and tools, Galaxy tools. And in this case, the use Galaxy.os and tabular learners tool is selected, and Gaia will run the analysis, run the tool for you, and presents the user with a complete schedule. And then the user can go directly to the Galaxy history to view the detailed outputs, like this HTML outputs. And a little bit more technical detail about Gaia. Gaia have 2 main parts, agent logic and the agent tools. The agent tools web Galaxy APIs, so the LM can use these agent tools to do to use this agent tool to learn do something like create histories in on a specific Galaxy instance, or upload the datasets to the histories, or run the tools on your behalf, something like that. And when a user sends a query, the agent logic will pass the query to the LM, and then LM will decide which agent tool it wants to use, and then the agent logic will run the tool for the LM. And the agent logic will also return the agent tool execution result back to the LM. So LM will, based on this execution results, to determine which Galaxy instance and which Galaxy tool it wants to use. And agent logic will run the Galaxy tools. We run the Galaxy tools using the runs Galaxy tool agent tools. And this setup allows Gaia to break down the compressed tests into a small piece, and then handle them step by step. To achieve this, we do use MCP, Model Context Protocols, to develop the agent tools. But the talk right after this is about the MCP tool for Galaxy, so I'm not going to discuss too much about the MCP here. And another important part of Gaia is the prompt. For Gaia, because we have a very clear workflows step by step. Like if we want to run a Galaxy tool on Galaxy, then first you need to create a histories, and then upload the datasets, and find the tools and run the tools. So we have a very clear workflow here. So what we provide to the LM is a step-by-step prompts to instruct the LM how to perform this test. And another very interesting lesson we have learned is that we have to tell the LM in the prompt, don't give up until you reach the final answer. Otherwise, it may stop as soon as it encounters the first error. And we know that for scientific products or software, the reproducibility is a very important feature. So using the agent tools and step-by-step prompts will definitely enhance the reproducibilities. And because it will minimize the chance of producing irrelevant output from LM. And while agents like Gaia's might run different agent tools, might run different Galaxy tools, for the same queries. But the Galaxy tool itself is still, reproducible. Like any jobs executed by the LLMs or the agents, you can go to the Galaxy history to rerun the jobs anytime you want. And tailways and some framework. Myself, also as a software tool developers, I would like to say that we can try to make our tools easier to use with clear descriptions. That not only benefits the agents like Gaia's, but also benefits the users. And the framework is not complete list, but there's something we would like to do. The first one is enable Gaia to transfer file directly from cloud storage to the Galaxy instance. And we're also trying to evaluate the best interface for the user. And for the Galaxy training tutorials, we are thinking to let GLM to use them to do the better recommendation. And acknowledgements, we still have 2 talks this afternoon in session 2A about Galaxy data analysis agents and Galaxy machine learning tools week. And because we take any questions, I would like to say that we probably only have 1 or 2 minutes for quick, 1 or 2 quick questions. But if we have any questions or comment suggestions, feel free to stop by our poster session. We can definitely talk. Yep.
Okay, so Junhao showed a really cool example of how we're using this right here, right? So I'll walk into a little more detail about how it works on the back end. There is some overlap in the motivation, so I may gloss over some of that since he did a good job of it just now. But anyway, so this is GalaxyMCP connecting LLMs to your Galaxy, right? So the clicker's not working. There we go. Okay, so the challenge, just like Junhao set up, Galaxy's huge, right? There's so many tools. There's tons of workflows. They're all over the place. All the servers are different. Some of the workflows are on this external IWC resource. Some are in your Galaxy. And the challenge, it's a big challenge for even someone who knows what's out there to navigate, right? So LLMs are really great at looking at large sets of data and summarizing or finding key points in them. So finding you the right workflow, that kind of thing. So it's like searching for a needle in a haystack, but you can just get the AI to do that for you. So the way modern LLMs work, right? So context is super important. A couple of years ago, I made a first pass at, we called it ChatGXY at the time. And the problem we kept running into was how do we get the right context to the LLM to actually do something useful, right? For some things, it was really easy. Like, okay, here's a tool error report. Given this error report and the fact that your LLM has been trained on basically everything that's been put on the internet, for better or worse, what do you think this error means? And it's actually really, really pretty good at things like that. So it can explain concepts that are fixed, like static stuff that's well documented, well explained. Hopefully everyone's writing code with it. But it can't see any of your stuff. It doesn't have your context. It doesn't know your data sets. It doesn't know what workflows are available to you on the IWC, if they've been updated, et cetera. So yeah, it's very generic advice. So context is super important. I was actually very similar to the demo we did yesterday. I was trying to set this up and show it to someone, and I forgot to turn on the MCP server. So this is what happens without context. Are we connected to my Galaxy? Oh, why, yes, we are connected to the Milky Way galaxy. We're all connected and, yada, 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 yada. Thanks, Claude. That's exactly what I wanted you to do. So where do we get the context? MCP is where we get that. It's the bridge between LLMs and, sort of whatever you want to plug into it, really. There are a couple of different things in the model context protocol. There are tools, there are resources, and there's one other thing. I'll look it up later. I would have known it 5 seconds ago too. So it's an open protocol. Anthropic came up with it, but they've open sourced it. Basically, everyone's implementing tool use at this point. So it's the standard that you would want to build things on. The key thing here is you can write it one time. We'll have a set of MCP servers for Galaxy that you can then use in whatever agent you want to build. Junhao's using it for Gaia. We have a little command line, agent. We'll eventually use it in the Galaxy interface, we'll see. But it enables AI to actually do things and not just guess based on what it knew from 5 months ago or whenever it was trained. So yeah, bridging the gap with the MCP server enabled, obviously, it can connect to my Galaxy and tell me a little bit about it and actually do things in real time. So the basic architecture here is, so there's a protocol and then all the rest of this is what happens in the MCP platform. It's a fast MCP2 server. It's written in Python. It basically uses BioBlend, which is a huge benefit here, right? We already had this great API library that we could just use and we didn't have to build again. So we use BioBlend to talk to Galaxy and then pass everything back. So currently, what can it do? Obviously connect to your various instances, can get basic information about it, about the server and your user, can do a bunch of different ways to search tools. I don't have to read all these, but so data management, uploading, downloading, monitoring execution details, so it can look at your workflow invocations. The other thing that's kind of important here is that we're pulling in not just Galaxy stuff. So it also supports talking to the IWC to find real-time, the most recently updated good workflows, right? So you can search the IWC for an RNA-seq workflow or for a multi-sample tissue, whatever we did yesterday workflow. And it actually works. So some use cases here, I imagine it's not just for someone that wants to talk to a natural language model. You could have multiple different use cases, right? So if you're a wet lab researcher that's not super computational, you just describe the samples you have. Help me do this. And it'll interpret that, look through the whole set of things to do, and guide you through the analysis. For a grad student that wants to know why their tool failed, right? That really helps, but it enables us to get the context from Galaxy of their tool that failed to the LLM and then do some work on it. And for a bioinformatician who really just needs help like searching faster, you can find all the right workflows and go. So it's all about translating natural language into Galaxy actions. I'm gonna just gloss over that for now. We saw, this is basically what I showed yesterday with how you can really just sort of, given this huge set of tools, you can let it go. And here, what I wanted to show is that I also had other MCP tools enabled where you could search the web. So you can imagine combining a whole bunch of these things. You could search the web, there's a file system MCP server, but basically you can take all of these tools and put them together and build something really cool where it's gonna look at your local file system and find your personal references and papers, or search the web and find, okay, Galaxy didn't have a citation for this tool. Let me see if I can find it or find more information about it. So you can see how it allows LLMs to grab all of the context and pull it all together for you. So technical implementation, yeah, it's fast MCP2. We've just updated that. BioBlend, Pydantic AI is what we're using for our little reference CLI implementation. It's like just a command line, talk to Galaxy in your terminal kind of thing. Yeah, it's well documented. That's kind of what writing a tool looks like. So it's very simple because we use BioBlend, but the Galaxy state is just sort of the connection that's maintained in the server so it can talk to your Galaxy. And then we literally just use BioBlend and say, hey, get the tools, show me this information. And all of these are sort of, they're curated to, you don't want to just plug an LLM into the whole Galaxy API, right? There's a ton of endpoints. There's a lot of crazy stuff it could do, and it's just overwhelming. You want to get just the right context into the LLM. So all of these sort of massage the request or the data that comes back to present it in a form that is useful. If you did want to try this now, it's really easy. So Claude Desktop is what I showed off yesterday and in some of these screenshots. It's a blob of JSON, but basically it's UVX, GalaxyMCP, and you have to, obviously you have to tell it which Galaxy you want to talk to, and you give it an API key, and you can run this now if you want. It's super easy to develop too, if you actually wanted to play with that side of it and see how, adding your own tools. And then, yeah, so where we see this going. Yeah, right now, so there's standalone MCP server. Other things are, either using it or in the sort of AI LLM working group. We have okay, so with the MCP server, right now it runs locally in your machine. So what we would like to do is have a hosted MCP with unified auth with Galaxy. So you log into Galaxy, it already knows who you are, and then so we can have requests made to that Galaxy on your behalf without having to run anything locally. And then once we have that, we can circle back to like ChatGXY that we tried to do a couple of years ago and really make it happen where you can have a natural language interface in Galaxy that then uses correctly contextualized MCP server to actually do things in your Galaxy in real time without having to sort through, fish through all of it yourself. Yeah, so that's the vision. And that's that. Perfect. I'd want exactly.
Yeah.
So like you mentioned that in your work also, and Junhao also used the ChatGPT and the application like Claude. When specifically you use that application for the basic purposes, it's free. But when you go little bit advanced like this, so like you have to pay for it. So do you want to comment, like, is there any way we can use DeepSeek, or you are?
You can, yes.
trying to use it?
So Jetstream does have a hosted inference service that we're actually going to use for usegalaxy.org. It has DeepSeek, the 671 billion parameter model, and Facebook's or Meta's Scout. And those are freely available if you have access to, if you have an allocation. And we would like to use that to bring all this straight into the Galaxy interface, so nobody has to use any API keys or anything, right? You're not paying for the LLM, Jetstream is. You can also, if you would like to play with it locally, the MCP servers, you can plug into any sort of agent you want, right? So there's the reference command line agent. You can just point that at, have you used Ollama? Look it up. It's OLLAMA. But you can actually run some of the admittedly worse models locally on your laptop if you just want to play with it with the MCP server. And depending on what you want to do, they're actually totally fine. It's not going to write your paper for you, but.
I was wondering if you've tested any of this in other languages. So I think it would be really cool, because right now, basically, when you get to a certain level of science, you kind of have to do it in English in very specialized fields. But I think it would be really cool if you could use this to make it more accessible. So somebody who's, say, just finished undergrad in their own language, maybe is just starting to learn English to get into graduate school, but could possibly use this to do analysis. So just wondering if you've thought about that angle and if you've tested it at all in other languages.
I haven't, and I only know the one, but that's an awesome idea. I mean, that's, yeah. I mean, that could be, so all of our training materials are in English, right? So you can imagine smoothing out that interface for someone that's not a native English speaker. That, that's a really cool idea. Now I want to go, I'm going to use Google Translate, which isn't a great translation, but I'll try it.
I just want to make a quick comment. So there are scientific language specific AI translators that are developed, and they're kind of like more from the documentation point of view, but there's no reason you couldn't apply them for something like this.
Cool.
Okay, so we'll move to the next presenter. So
Yep. So good morning, everyone. My name is Jayadev Joshi, and I'm a research associate working at the Cleveland Clinic with Dr. Daniel Blankenberg. And thanks a lot for GBCC team to give me this opportunity to present the work in front of you, and thank you everyone to be here. In this talk, I'm going to talk about the automated integration of R and Bioconductor tool into Galaxy. So the background and specifically the motivation of this project is that we want to simplify the tool integration inside the Galaxy. Specifically, if you have some Python or R-based tool. Specifically, in this talk, I'm going to talk about the tool if you have R-based or the Bioconductor-based tool. So the biggest motivation of this project actually come from similar surroundings. So when you look like across the different packages and the analysis tools are available, so there are huge amount of resources available. But when specifically you want to assess like how many people actually can efficiently use those tools. So due to the lack of programming experiences or the hesitation to use this advanced approaches, you always feel an unmet need to this kind of research area where you have to come up with certain solution that actually can help the wide range of researcher who are actually not very experienced in the programming and computational sides. So with this, I'm going to talk little bit about the Bioconductor and Galaxy, although this 2-3 days we are going to talk more and more about these platforms. So like specifically when you talk about the Bioconductor, so it's mainly built on R. And like as of the release 3.21, it's there are more than 2,300 tools are available. And it offers the cutting edge tool for the analysis and visualization. When it's come to the Galaxy platform, it's excellent browser-based tool, and like it offers like thousands of tools that you can install, and also you can use on the pre-built resources, and it supports millions of job like every month. So the biggest issue, like specifically with those who are not very much experienced with the different programming languages, is to it's a little bit steep learning curve when you want to integrate this tool inside the Galaxy. Also, when you have large amount of tool you want to integrate, you have to do so much manual stuff. So we want to automate this stuff so that it can address these two issues actually. So the solution of this problem is the R2G2 package. So R2G2 package actually try to automate it, try to help to generate the Galaxy wrappers automatically, and it also try to manage the dependency, and you don't need to have any kind of programming experience. It provides a simple user-friendly command line interface. So I just want to give a quick background, like how exactly the Galaxy tool implemented. So Galaxy tool has two major components. The first is either a Python or R script or any script in any other languages. And the second most important component is the XML wrapper, in which actually you try to implement the different GUI component of the tool. So when you combine both these tools, this GUI can actually execute your job. So with this, so how exactly the R2G2 work, and it generate the XML wrappers for you. So right now we have two approaches. So in a first case, like if you want to generate the tool, you don't have any kind of R script, but you have a Bioconductor package, and if you want to generate tools based on that, so so R2G2 actually iterate over all the object of that package. It try to determine all the available functions, and it extract all the information and the function definition. Based on that, it actually create the essential component of an XML wrapper Uh, yeah, so it actually generates the command line interface for your tool, the input parameters option and the output parameter option, and eventually it combines everything into a Galaxy tool wrapper. In case of the second approach, if you have an R script with the argument parsing instruction defined, so it also can utilize that, and we try to implement a reverse engineer argument parsing approach, which actually inherits some of the components from the argument parsing library of R, and it actually generates all the argument parsing instruction. It convert into a JSON format, and then it's convert back to a Python compatible argument parsing, and based on that, actually it generate all these three component again. So in summary, if you have like any bioconductor package or the script, you can easily generate your tool with this. When we talk about the availability is already available on the GitHub, and we are planning to release the Conda package also for it. And it has some limitation in terms of like how many tool it can generate while you are iterating over the packages. And also we are trying to implement more support for the different argument parsing method. With this, I just want to acknowledge my team at the Cleveland Clinic and funding sources, and especially thank to the GBCC team for the scholarship award. Thank you.
Okay, so we can have one question.
Hi, thank you. Great talk. I have a question about, have you looked into the scale of the different tools that you think would fit into this model? And if you have looked into how many tools would fit into this model, what is that number on average?
So if I understand correctly, you are asking about the complexity of the tool, which has, or the scale of the tool. So if you have like any number of I mean scripts, all the packages, so you can use our G2 on that. Yeah. So
Thank you.
Yeah. So I think this ties in actually very nicely with the previous talk. Yeah. So I think like one of the main limitations in adaptation of Galaxy is that we have a lot of cool things. You can get started very quickly. We just heard how many awesome tools we have, and they are like written with a lot of care and maintained by a passionate community that really wants you to use computational tools in an easy and digestible way. And I think it's only fitting that, like, as the spirit is, that like we want you to be able to do everything you need to do, right? So if you want to run package XYZ and it's not in Galaxy, like, well, that's a limiting thing, right? You feel like, okay, I learned all this stuff, but like now what, right? And you know, why do we not allow people to install tools, right? Because you can already run code in RStudio, as we saw yesterday, and we also have Jupyter, and in principle, any sort of thing, we can fire up a shell. Why are we not allowing you to do that, right? And like, it's 2025, we figured out containers and storage and compute, so it would surely be possible. So yeah, why? So there's one key thing that is holding us back. It's also served us really well, right? Like this is how Galaxy tools look for the last 18 years. And what I want you to focus on is what I highlighted in this little red box. So you see the hashtag for dataset in datasets, and then the N4, and inside you have like $dataset. So what this does is it iterates over the datasets that are defined here in this particular input, and then it's just gonna place the string representation of the path in there, which is a lot of things to glance from this if you're not familiar with it, right? There's a lot going on. And this seems relatively benign. However, this is a whole sort of programming language embedded there that is based on Python. And you can import anything that is available in your current environment. So for instance, you can import whatever is available. So as an example, I imported pathlib path and opened a file on disk and wrote to it. Or we have this particular object that is like Galaxy's God object. That's the database. So you can query things in the database from within the tool. Like, absolutely don't do this, please. But you know, if we allow you as a user to bring that tool, you can do this, and we don't want that, right? We run a service for everyone. There are certain things a user shouldn't be able to do because that also has nothing to do with running a tool. So how do we have this many tools if that's such a problem? So there's many GitHub repositories maintained by the community over the years. And we have a review process, we have a testing process, experts are looking at every tool and ultimately placing some trust on publishers like the IUC or GalaxyMCP or imaging tools, or, you know, and then as an admin, you say, either I trust my users anyway, so I don't care that they'll be careful, or you read all the sources, or you just trust that we were careful enough here. But even for us, this is a mistake. Like, things can slip through and then we'll remove them. But, you know, why not make it better? So there is a straightforward thing, and we sort of like lifted this from CWL. So instead of a cheetah template, we have a JavaScript expression in here. So this still turns into a string that ultimately lists all the paths to the data set here and writes them out to output.txt. Also note this is YAML. I think people like writing YAML much more. And then what we do is, instead of giving you access to everything Galaxy has access to, we just give you JSON. We just give you a JSON object with the particular things filled in for the run. So this is already safe, but we're back to a problem, like, you've got to know what to use there. And if we're going to change how Galaxy tools are written, it must be better than what we had before. So we can do something really cool, which is define a schema for what is available here. So that when you hover over anything within these expressions, it can tell you what this is. So now I'm hovering over input, and I can see that these are all the attributes that are available and that I can use. And if I go down one level further and I just hover over path, we have even description of what each thing is. So like, as you write, you can learn what's available. You can press tab and you see what sort of things could I potentially be using. Because we build up this TypeScript interface for the inputs, it can also catch things like typos, which that's a bit of a contrived example for the sake of time, but like, here there's an A missing and it says like, hey, did you mean that sets? Which, no, actually this is the typo, but still pretty good, right? So what's happening behind the scenes? I don't have time for that. So this is the schema. Doesn't really matter. It's a beta feature, so you'll not yet see this on usegalaxy.org unless you ask us to enable it for you. And I think we're still not saying yes to that question, but it's going to come. This is how you do it if you're an admin. They are private to the creator so that we don't pollute the whole list of tools. And it's of course like a social shift in that like people can bring their own tools, and of course they can write broken tools. That we didn't solve there. We made it a little bit easier to write good tools. But like if you're running a server like usegalaxy.org that over time had like 300,000 registered users, you can't have every one of them contact you for a broken tool because like there is now not that central source that you can just fix. So it doesn't obviously solve all the problems, but can't always aim for that. There are a bunch of limitations that we're going to solve eventually. So yeah, I mean, that's a feature that should be coming to a server near you. Also, if you were paying attention yesterday to this huge success of the live demo, that you saw how to use it. So like there's this little extra thing here if you have the correct permissions. And then you can click on the edit button. Monaco, which is this editor, pops out and you can start typing, or you can edit an existing tool. You press save, and then if you don't click on the edit button, you get to the tool form. We'll improve this over time. I'm not really a front-end developer. That's the best I could come up with. So with that, I want to say special thanks to John Chilton, who did the modeling work of the schema based on like a discussion we had at GCC 2022. And then it has percolated over time into something hugely useful that we didn't anticipate we'd use at the time. Michael, who was pushing us to allow users to come with tools, because we're also working on CWL integration, and like if you can write your CWL workflow, but you still can't get the tools, that's relatively not so useful. And then Nicola, who is always a great help with just reviewing code. Yeah, so thank you.
Okay, so we can have one or two questions. So,
and I think it would like perfectly integrate with what you're writing, because there's an API to it. Like you don't have to use the editor. You can use whatever. You could even imagine an interactive bash system where you just write what you're doing, and then it like behind the scenes uploads the tool and runs it.
Hi. I had this question last night while you were doing the live demo as well. I see that there's a path parameter there, which is not an input. So in the shell command, it's ln-s inputs.input1.path, while inputs only has the two keys, name and type. I think the path comes from the schema.
Right. So that's the object. I think I could probably talk about it for an hour. Yeah, so it's coming from there. So this is like the real core of it, where we're putting in this defined structure for the data sets, and that's in fact what is there. That's where we take it from. And what you see on the left is the abstract definition of what the tool looks like. But if you would say it's type data, then it knows it needs to put this in. It would look very different if the type was integer or float. Then you wouldn't have a path, base name, root, blah, 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 because it doesn't make sense. And because of this integration with the type hinting, it would also tell you that that doesn't work. Like,
yeah, that was my question. I was like, where is that coming from, and how do we make sure that we don't put the wrong type in? Thank you.
Yeah.
Okay, so in the next 10 minutes, I will tell you everything you need to know about Galaxy's new visualization framework, from its motivation to its design. And many of the elements were already mentioned by Sergei earlier today, so I'm pretty happy about it. There is some overlap apparently in this logic. And let's jump right into this. So we overhauled the entire visualization framework, and we migrated all the visualizations to this concept, right? And most people, when they hear, oh, great, another framework, right? Something new to learn, but just hold on with that for a minute. So our goal here with the motivation is we want to design a JavaScript framework and we want to uphold Galaxy's core values at the same time. So that's the main challenge. So what are the Galaxy's core values? It's like reproducibility, particularly. That's why it's the number one, the most important one. When you run something today and run it 10 years later, you want to be able to reproduce the same results without having trouble. And the other core values, of course, accessibility. It has to be easy, accessible through the web interface, and transparency for the user so he knows exactly what is actually happening. How does this visualization, in this context of visualizations, how was it generated? And this becomes particularly difficult if we look at the development and the popularity of popular JavaScript frameworks. So here you see a ranking of popularity for the time between 2014 and 2019 of popular frameworks. And previously, we always had the discussion with a certain focus on picking one and focusing on that. But this makes it very difficult. How do you combine reproducibility over decades if the development is so fast? So we took a step back and dramatically simplified the interface and Galaxy's role. When Galaxy hands over from the interface to a visualization plugin, this is what it populates: a simple HTML template. And it contains three elements. There is a container with the ID app. The ID can be customized, but default is app, and for our case, it has been sufficient so far. And there it attaches an incoming object. This incoming object contains at least two things at this moment. This is a root attribute, tells the visualization where Galaxy is, basically the host URL, and a dataset ID, the dataset the user has selected to be visualized. It can contain many other settings and options, custom to your visualization if they are saved, for example. And all of this is documented. I will get to this later. But the other two elements are a style sheet developed or provided by the developer of the visualization and just the script module. So this is the complete interface. Every interaction from the visualization back to the Galaxy server has to go through the API. So this interface allows us to have a very simple, minimal example here. So you have to have an XML for your visualization with a name, description, and here you have a script defined, and this is a minimal script. So what does this minimal script do? Once it runs in this template, the one you saw earlier, it can access the app container and it can access the attributes provided by Galaxy. And after that, it's on its own, right? Regardless what it does. So this is the first step for this to ensure reproducibility. But we have to go one step further. And there was a question regarding what happens if packages changes and so on. So what we do is we use NPM, right? Obviously, it's the largest package manager at this point. And we have a requirement. The published bundles are static, so Galaxy doesn't do anything with it, just loads it. So you pre-build everything with all your dependencies and push it statically to be published by NPM. And Galaxy just, there's a YAML entry, you can add this package, and that's it. So that makes it versioned and immutable. It's a fully standalone application. So it runs not depending on Galaxy, it runs with Galaxy. So you can run these visualizations anywhere. You don't need Galaxy for them to run. But if you want to access data, use the API, you have to point to a Galaxy server. So this separation also allows us to ensure that there are only a few remaining risks left, and those were actually mentioned by Sergei earlier, that in our case, the Galaxy API could change, which we have to keep an eye on. But the other thing is just loss of browser support, which is very unlikely. So as long as you can point to a CDN and provide these attributes, the visualization will run because it just runs in the browser and looks for Galaxy just from an API point of view. So that means you can use any JavaScript stack, right? Whatever you like. Also WASM, and I will come to an example for that. So now we haven't done much in some principle. We just have a very simple interface, and now the developer is on its own, right? So what we do to remedy this a little bit, we provide, for example, a view package for you. If you develop your visualization, you have an XML for Galaxy. In this XML, you can specify input parameters like you do for tools. And we provide an optional view package to render the visualization tool form, basically. So we keep the interface simple. And on the other side, we provide packages or configurations for you to assist you with the development of your visualization. If you need them, if you prefer them, if you want to do the input elements yourself, that's perfectly fine too. But this, of course, helps because once these inputs are in the XML, Galaxy knows about it. If you use them in your workflow reports, they will directly appear there, and you can link your data to it. But again, this also runs separate of Galaxy and just interacts through the API. So we had named this Galaxy Charts because it was a progressing framework, but maybe it should just be called the Galaxy Visualization Framework because it's the only framework we use at this point. And it's the only one we'll need. And everything is documented, every detail at this URL here. So everything means there are examples to copy paste. There are different variations, some utilities. It shows you all the inputs are listed for you to like just copy and paste, basically. It shows you how to connect to a Galaxy server. It gives you detailed backgrounds on the entire framework with some additional options. There shouldn't be anything missing, and I think it takes about maybe half an hour to read all this very carefully, if needed, to have really all the details. There are some Vite configurations if you want to start from scratch. How do you read but the minimal example I showed is actually pretty explanatory. So we added, as I said, now 35. It's pretty, it's a good number, but hopefully we can grow it. All the visualizations can be transferred. You see here in the Plotly example, this form was automatically generated from the view package we provide. So there's a standardized look to it if you want to use that view package to parameterize your visualizations. We also provide now example data sets for every visualization. So when you click on a visualization, it will, maybe see this in a second again in this GIF, it will switch over and show you help text as markdown, and it will allow you to upload example files so you can test all the visualizations however you like. So there's one particular one I want to picked out. It's Jupyter Lite. So that's a Pyodide-based one. And again, it's fully packaged. It runs anywhere. And we can just link it or load it into Galaxy, right, like here. And then we provide a Python package so you can access your history data sets or write back to it, right? So the interaction here. And some packages are pre-installed, like here the Plotly, NumPy, for you to get started right away. But you can install pretty much any package there. And one example here is how to test this. Since the visualizations run entirely independently, you can develop them without Galaxy. You don't need to start up a Galaxy. If you want Galaxy data, you have to point to one. But what we see here is the automated testing for this Jupyter Lite visualization, which runs separately. And the only thing mocked at this point is the Galaxy API. So with that, this, of course, is a huge community effort. Many people are involved in this, and we successfully transferred all of this. We'll have above for more questions, and I will also give a demo for everyone who's interested. Thank you.
Okay, hi, good morning, everyone. My name is Leandro Laiborio, and I work in the Scientific Computing Department of the Science and Technologies Facilities Council of the United Kingdom, the STFC. And in this presentation, I would like to show you the work that we have been doing on developing an end-to-end simulation system for one of our latest facilities that we have at the STFC, the so-called Extreme Photonics Applications Center, or EPAC. I think this presentation is going to be a little bit different from the previous ones, because here we are using Galaxy for managing a workflow that it's associated to a large-scale complex materials science experiment. So we have no biological samples here. I think if you put one in front of this laser, it would fry it instantaneously. So, and that's why I decided to structure this talk like this. First, I'm going to briefly tell you what is a large-scale experiment in a national facility and why that large-scale experiment is different from something like a new facility like EPAC. Then I'm going to tell you briefly how EPAC works. EPAC is a laser accelerator. I'm going to give you a few clues about how these machines work. Then I'm going to tell you about VPAC, which is the name we've chosen for the end-to-end simulation system that we are implementing in Galaxy. And then I'm going to say a few words about why we think that Galaxy is a kind of an ideal platform for developing something like VPAC. And then I'm going to go to the conclusions and tell you a little bit about our plans for the future. So what is a national facility and why EPAC is a particular type of national facility? This is an aerial picture of the Rutherford Laboratory at STFC in the United Kingdom. This is where I work. This massive circular structure in blue is Diamond, the UK synchrotron. And with this machine, you can produce very stable X-rays that you can channel through beamlines that we have that are managed by staff in the lab. And then, as I said, these X-rays are very well defined, clear energy, and each beamline has a specific experimental setup depending on what you want to do. And as you can see, this normal accelerator is a massive structure compared to a laser accelerator, Astra Gemini. This is the same type of accelerator as EPAC. And also these things are much more expensive. But it's not the price and the size the only difference. One of the main differences between a laser accelerator and a standard accelerator is the fact that laser accelerators like EPAC are flexible. So these are machines that you can adapt depending on the sort of radiation that you want to generate. For example, we can generate the entire X-ray spectra, and we can even generate gamma rays. And it is this flexibility what makes being able to model, track, and record the whole process very important. And that's why we are building this end-to-end simulation system for EPAC. And what is EPAC? How does a laser accelerator works? Okay, this is a schematic view of the experimental area 1 of EPAC, which is the one that we are simulating. Basically, you have first a very strong laser pulse that goes into the system and goes into an optic system. And inside there, you can process the laser pulse. You can filter the pulse, you can change its intensity. And once you are happy with your pulse, you fire it into a gas chamber that contains a gas of known density and composition. And when a high energy pulse goes through this gas, what happens is Wakefield acceleration. It's a phenomenon where the electrons are ripped apart from the nucleus of the atom. You generate the plasma, a gas of ions, and then you accelerate the electrons. And because of that acceleration, you generate radiation. And you use that radiation to do the experiments that you want to do. You can do X-ray imaging or X-ray absorption spectroscopy. It depends on what you want to do. So that's how EPAC works. And as I said, we are building VPAC, which is an end-to-end simulation system for this laser accelerator. It comprises essentially 4 software tools that once you connect them, you can model the entire process of EPAC. And this is a schematic view of this system. You have basically 4 tools. You have a tool for modeling the gas target, a tool for modeling the laser pulse, a tool for modeling what happens when the laser pulse goes through the gas and generates the plasma, and then another tool to manage the radiation that comes out of this plasma and to do the X-ray absorption spectroscopy experiment or X-ray imaging that you want to do. And as you can see, this workflow have feedback loops, and you can tailor your process until you are happy, and then you can model the whole thing. And then once you have your 4 modeling tools, you can connect them in a workflow that will help you simulate your end-to-end simulation system. And here you can see the name. So we have CFD for the gas target, LACY for the laser pulse, FVPIC for simulating the generation of plasma when the laser pulse goes through the gas. And then we have GAN4, which is a tool developed by CERN. We are using several modules of that tool to model the transport and the use of the radiation for doing your experiments. And essentially what we are doing is we're generating associated Galaxy tools for each one of these tools. And then we're going to connect them in a Galaxy workflows and run the whole simulation system. And why we think Galaxy is a good thing, or is an ideal system for modeling these sort of experiments? Well, essentially because Galaxy is a web-based graphical user interface. So from the user point of view, you don't have to worry about installing anything or maintaining anything. Also because Galaxy workflows are simpler than standard workflows, and you can run them in one click. Also because we can control the computational power of Galaxy. So we can run different tools in different places depending on how computationally expensive they are. These two are very expensive, and we are running this one in a graphical processing unit, for example. And then perhaps another key thing is this thing that Galaxy has that is not common in other workflow systems, which is you can change the inputs of any of the tools and then leave the rest of the workflow fixed and then propagate this change to the end with one click. This is very useful for what we want to do here, because basically we have three key applications for this end-to-end simulation system. We want to use it for the design and testing of different EPAC features. We want to use it also for optimizing specific operation conditions. So a user comes and wants to set up the machine in a specific way. And also we want to use it for testing potential upgrades for EPAC. So these are all reasons why we think Galaxy is very useful. But there is another one. Everything that you do in Galaxy is transparent and reproducible. So imagine you set up your machine and you play with a lot of parameters and you are happy with the end result. Then you can save everything, the workflows, datasets, parameters, full history into a digital object like an arrow crate. Then you can put this arrow crate in a database like Zenodo, attach a DOI to it if you want, and then you can reproduce what you did exactly. And this is crucial for these new types of facility. We want to have a database of these things and be able to run things whenever we want. So that's it. So the conclusions are, I think I've presented you EPAC, which is a new type of large-scale facility that essentially is a flexible facility. You can adapt it to do what you want to do. And with this thing, you can do different types of experiments, X-ray absorption spectroscopy, imaging, and these experiments are compatible with the ones you can do in the big accelerators in the national facilities. And we are developing VPAC here, which is an end-to-end simulation system that allows you to model, track, and record the whole process of VPAC. And VPAC is being implemented in Galaxy because, well, it has simpler workflows. It's a web-based graphical user interface. You can efficiently manage your resources. It's optimum for design and testing, system optimization and upgrading, and most importantly, it's fully transparent and reproducible. This is a work in collaboration between the Central Laser Facility and the Scientific Computing Department. There is a lot of people behind that. I'd like to thank them all, and thank you for your attention.
I feel like I'm drowning, to be honest with you. The amount of new technology and cutting edge approaches to computation and analysis that have emerged up to this point, very early on in the conference, is pretty astonishing and overwhelming to me. What I'm going to talk about here is something which I think is really important for building the workforce, building the student capabilities for moving forward in modern genomics. And it's the idea of producing libraries of well-characterized, exemplary experiments that you can actually do work on, reproduce earlier analyses, and figure out how to make them better And to do this in a way that is more accessible to more computational frameworks than we have accomplished thus far. And so I'm going to be talking about work that is done mainly by Aaron Lund, who's been a longtime contributor to Bioconductor at Genentech. And I don't know, I'm not seeing this on this screen here, so I'll just look at the screen here. This is something called ArtifactDB. And it's capable of doing the thing that I just described, which is producing a library of analysis-ready artifacts. Is that how they put it there? Yeah. So you don't have to do a lot of work in order to start computing on experimental results from a great many paradigms. Single-cell RNA-seq, multiomics, proteomics, and so forth. And the tooling underneath it already exists in a number of Bioconductor packages and Python packages. And what I'll talk about is the example of a spatial transcriptomic experiment, the serialization layout, schemas, validation, and the idea of a data service underlying this, so that from whatever language and framework you may be working, you can issue well-defined API queries to recover data into your system and perform analysis. So here's an example of a spatial experiment. There are 50 features on 99 cells, and the features are identified with ensemble, mouse gene identifiers, and there are cell-specific column data characterizations. Where in the array did they live? What's the sample ID? What tissue is it from, and so forth. And then there are spatial coordinates and image data available on this. And so that's the our Bioconductor structure, right? And that's been influential in genomics. Structures like this are also available in Python and data, for example, or spatial data. But this is R, right? I mean, there's a lot of RDS that when you try to save this object using save HDF5 summarized experiment, for example, you get things that are more native to R and to Bioconductor. And we want to get away from that. And so here is a representation. If you use the language agnostic approaches in Alabaster to save this object, you're going to get a metadata structure in JSON, and then a tree structure with all the different files that are being used. Some of them are using HDF5, some of them are using image representations in PNG, and so forth. And this is completely language agnostic in the sense that there's just JSON and there are folders with these things that are usable in any language. And here is the top level object metadata. And you can see a lot of attention devoted to versioning. And the idea of the ArtifactDB is that there really will be an efficient archaeology of any given data artifact as revisions are made and so forth. They're all accessible through the API. And this is what the mapping H5 looks like. And so alabaster.spatial takes care of save object for these spatial experiment representations, and read object will materialize them back into a spatial experiment in R. And if you need to do it in Python, then the same data structure in those folders could be brought back through the Dolomite package. And this is one that, again, goes back to work of Lambda Moses in the spatial feature experiment and the Voyager packages. I really needed this for the work that I was doing that presented in the live demo, because I needed to be able to take the post-analysis spatial feature experiment where the different Moran and Geddes statistics are bound into the object. And I needed to put that down in a disk format that I could zip up and take to wherever Galaxy was running and then load it back in. And that worked very well. So there's an alabaster.sfe package that takes care of this problem. And then the validation should be something that's very efficient, and it's all done in C++, and that's taken care of by these libraries that are due to Aaron, again, in a package now called a sorthead to verify the importability of these artifacts in both R and Python. And so more serializations are developed for R than in Python, but they are still growing in both ecosystems. And the one thing I wanted to emphasize for the teaching and learning of single-cell RNA-seq is the scRNA-seq package, which now is usable in both R and Python, drawing from the same exact data artifacts in ArtifactDB, but producing, in this case, 82 different experiments that have been published and are now reanalyzable by any student in Python or in R with the same package name, scRNA-seq. And the provenance metadata for all of these packages is available with the package, all these experiments. And this is what the single-cell experiment looks like in Python. It's different, but has the same basic functional capabilities as the single-cell experiment in Bioconductor. And so the conclusions are these very heterogeneous data structures for which we have developed patterns of work and documentation and so forth in R can now be serialized in a language agnostic way for use in any language that is capable of reading JSON and HDF5 and Parquet and so forth. Save HDF5 summarized experiment has a long history, and it mixes RDS and HDF5, but Alabaster Save Object goes beyond this to make something that doesn't use RDS, but can be used from any language. And then the Gypsum service is an interface to the ArtifactDB provided to make use of this in Bioconductor. So I hope that makes sense and is of interest to you, and I'll take any questions now. Yes, Casper. You have to take this item here, microphone.
So can you embed documentation inside of this, right? So this is sort of a serialization. That's great, right? But we also had a history that was sort of abandoned a little bit, but like also having self-documented objects, like where you have, description of things like.
That's an interesting observation. So one place you could embed provenance information in one of these objects is the metadata slot, right? And that can be done, and the contents of the metadata slot can be serialized. In fact, we ran into a problem with that because one of the pieces in one of the metadata slots, I think it was for the spatial feature experiment, was the version of the package. And the package version object is very special, and it didn't have a serialization method associated with it. That's been fixed. So you can put quite a bit of additional information in the metadata slot. We may want to build schemas for that and make it more formal, but I think that can be solved in this space.
Okay, we can have one more.
Yeah. So I was kind of wondering about the granularity that you might be interested in. So like one of the problems if you're running like long workflows is having to think about checkpointing, right? And having to like debug something that's crashed, and you're wondering like, well, this ran for several hours or days, and why did it crash? And so, it'll be like, have you thought about like trying to save intermediate objects and trying to like resume a run and
using this? Yeah, I haven't thought about that, but it seems like a very feasible thing to do.
Okay, yeah, hi, thanks for the introduction. I'm going to tell you what we did in the last 1 year, roughly, to streamline image analysis in Galaxy and make it more reproducible. Yeah, so the image community in Galaxy, it has developed gradually over the last few years. But if you want to designate specific birthday, you would probably pick the Galaxy User Conference of March 2018 in Freiburg, Germany, which was the first time that image analysis in Galaxy was introduced to a wider audience. In the next years, yeah, our major efforts were usually focused around hackathon events like the Biohackathon Europe, which is annually organized by Elixir and will also be this year. And if you want to get in touch with us, exchange, discuss things, stuff like that, feel free to join our monthly calls, which are on the third Wednesday each month at 4 p.m. Central European Time. So first of all, I'm going to tell you something about interoperability, because if you want to build workflows like workflows for image analysis, one tool that is hugely important, of course, is that your tools are actually compatible with each other. And we had that use case, which was about quantification of GL bands on Western blots. This was a protocol for ImageJ, which we wanted to translate to Galaxy. And to do that, we needed to manually annotate image regions of interest, which correspond to the individual GL bands. And this can be done in Galaxy using an interactive tool, like, for example, QPath. And we also have a tool in Galaxy for subsequent quantification of image intensities in the annotated image regions. It's somewhat blurry here. I don't know why this is. It looks like it's out of focus or something. Never mind, it's not that important. The important thing is that that tool, as an example, that requires that the annotated image regions are supplied as a label map, as an additional file. And in this file, the individual image regions should be labeled with with a unique image value, like intensity value, which identifies the corresponding image region. Unfortunately, QPath is not capable of producing such a file format for the output, for the export of the image regions, and this is why we came up with the idea to implement some support for the conversion of a GeoJSON file, which can be produced by QPath, to these label maps, which a vast majority of tools for image analysis is capable of using. And yes, this was done by Pavan. Much thanks to him for actually doing this. And another building block of our efforts last year have been to, of course, to bring new tools for image analysis to Galaxy, and also to incorporate major updates. Like, for example, among the newly added tools, we have added a tool which can split images along the image axis, which is very useful. For example, if you have a multi-channel image, but you have a tool that only can cope with single channel images. What you can then do is to split the image along the channel axis, so it will obtain a list of single channel images, which can then be processed by a tool. We have added a tool for background removal, which performs some background subtraction, which is a very common operation in microscopy image analysis. For example, we have added a tool for cropping images. Now we have it. And we have done we have added many tools to the Miro tool suite, which I will come back to talk to in a moment. We have also added a tool for segmentation of densely packed 3D volumes, which is PlantSeg. And finally, we have a tool for, which is basically called Ultralytics. This is a tool for processing images using YOLO models. It can also be used for training YOLO models. Very useful. Much thanks to Yisun for doing this. And on the update side, we have majorly reworked, for example, a tool for color deconvolution. We have updated a tool for processing of images using bioimage.io models. And we have updated SuperDSM to the newest version, which hugely improves the efficiency. And we have performed an update of the CellPose tool for reproducibility, and this I will talk about now. So CellPose, if you don't know, it's a method for cell segmentation, which is based on a deep convolutional neural network, which performs some prediction, which is called a flow field. And this flow field is like a vector field. This means that for each image pixel, we have a vector that points to, ideally, if the segmentation is good, to the center of the corresponding object. And then in a subsequent step, what CellPose does is it's called following the flows. It basically groups the image pixels which agrees upon the same center of an object. And this is how it obtains the final segmentation masks. So CellPose is nowadays widely used in image analysis. However, the neural network predictions can, of course, only be as good as training data that was used for training the network. So in case of CellPose, it was more than 1,000 hand-labeled images. And even though doing predictions using the already trained model is, of course, quite fast, one has to keep in mind that this is something that needs to be done, especially if you want to use more diverse data, then the training and the energy that is required becomes even more. And another issue that we had with the CellPOS integration in Galaxy was a reproducibility issue, and this goes as follows. So our CellPOS integration is built on the CellPOS Python module, and this module is it resides inside the Docker container, and inside the Docker container there's NumPy, which is built against the Intel math kernel library. It's also widely known as MKL, and on Galaxy Europe, we have containers which run on virtual machines. So I guess it can be different on different Galaxy instance depending how it is configured. Maybe it runs on bare metal, but in case of Galaxy Europe, it runs on virtual machines. And what we observed on Galaxy Europe was that we obtained inconsistent results for each run of the tool. And if you look at the predicted flow fields, you can see that the deviation is really strong. And what was even stranger is that when we tried running the Docker container without the virtualization, but on bare metal, we consistently obtained result A, which is the correct result. At some point, we realized that when we limit the number of threads which MKL uses internally to run the computations, to do its matrix multiplications and so on, to 1, so basically disable concurrencies, we consistently obtain result A on in all cases, in all runs, even in virtualization. So, and this leads to the question whether there is a concurrency issue in MKL. Apparently, it's difficult to tell. We can't tell ultimately. It might as well be an issue with the containerization, the virtualization, the hardware maybe, or some weird interplay of all these actors. But what we know for sure is that if you use MKL in your Galaxy tools, be careful and better double check whether things are going reproducibly, or maybe you need to turn off concurrency for MKL for some reason. To further enhance the robustness of image analysis tools in Galaxy, we have enhanced the support for the TIFF file format. So this is quite challenging. Supporting TIFFs is actually quite challenging because of the diversity of this format. So for example, a single channel image which comes as a TIFF file can come with or without channel information. Yeah. And we have updated our tools, so these cases will be treated more robustly. We have also enhanced the metadata, which is available in Galaxy for images. This now includes, for example, the image width, the image height, and the number of channels. This means that you can actually use constraints in your Galaxy tools or workflows that require that, for example, an input is a single channel image or a multi-channel image. Of course, there have also been plenty of bug fixes. Like, for example, we have fixed this tool for inference using bioimage.io models, which now also properly supports 3D images, much thanks to Anoop for doing this. And coming back to this Omiro integration. So for those of you who don't know, Omiro is a system for management of image data and the associated metadata. In a typical environment, you have there are some images acquired, and then they're uploaded into Omiro, and then you want to perform some analysis using Galaxy, and you download the images from Omiro into your Galaxy environment. Now, what is new that you can actually upload the results from that analysis back to Omiro, which makes the whole workflow much more seamless, and this is why it's quite important. And we have updated, we have done a lot of work to improve our training material. So we have written 6 new tutorials and updated 3. And I don't want to go through all of them, but just point out that especially this one is maybe quite cool because it showcases how a single workflow, which is for Voronoi segmentation, can be used for 3 different imaging modalities. In this case, it was for cell nuclei, for tree crown images from Earth observation, and for astronomy images. And to improve our outreach, so our community, the image community, is now represented on the Galaxy community board as a special interest group. And yeah, in the spring of this year, we had like a marathon of webinar contributions. So we did 3 webinar contributions for different organizations with an international audience. This was the Glow Bias Bioimage Analysis seminar series. This was Embly BI course on microscopy data analysis, machine learning and data bioimage archive. And these were the Eurobioimaging Image Data Community Days, where we contributed webinars on introduction, how to perform image analysis in Galaxy, how to use workflows, and how to use Omiro, and also how to use interactive tools. Okay, yeah, thanks a lot for your attention.
Super. Greetings. And it's really great to see you all. I'm happy to be here. I'm really happy to present to you some of the latest on NHGRI Anvil. And I don't think that I need to spend any time talking to this group about how the cost of sequencing has come down. Several orders of magnitude from $100,000 to roughly at about $1,000 in 2022. And of course, this has led to an explosion of population scale genomic data sequencing, genomic sequencing. And so now, today, we regularly see publications in the tens or hundreds of thousands or even beyond of a number of genomes being analyzed at once. So this provides some really unique challenges for the Anvil platform. The first and foremost is these are human genomic data sets. This is incredibly imperative that we maintain compliance with NIH genomic data sharing policy, that we uphold consent of donors if they want to be use their samples to research on one project and not another. We have to follow through with that. We need to provide very fine-grained access controls to maintain these secure and responsible data use. But of course, there's a tension at play here, and that's with trying to create a platform that's open, collaborative, and reproducible, something that the crowd here worked really, really hard at doing. And so, of course, we want to encourage sharing of data. We want to promote reproducible workflows and portable analysis and continue to drive interoperability with other platforms. And so Anvil is a bit of a balancing act here to maintain a security perimeter that is strong enough to protect these sensitive data, but also open enough to allow for biomedical data discovery. So yeah, this balance is really complex, but communities like you all, Galaxy and Bioconductor, are really lighting the path, helping us champion fair, open, and reproducible data science within secure environments. So why Anvil? Why do we want to do this? What is the whole point? What do we get out of the Anvil? And one thing that I really want to point out is there's scalable cloud infrastructure. So you can acquire your compute and storage as needed, scale it up for big analysis, ramp it down when you're done. In the case of Galaxy, we've heard about tools, user-specific tools being of import to some individual analysts. And of course, in Galaxy on Anvil, you are the systems admin for your own environment. You need a tool, you just get to add it. Bioconductor is really working really hard to make sure that the latest and greatest R and Bioconductor releases are available with precompiled binaries to make sure everything is snappy and zippy. And so really what we want you to think of Anvil as an integrated collaboration hub or a one-stop shop to access your data, run the latest tools, and collaborate seamlessly across institutions. So here's just like a couple of things that are going on in Anvil today. One of the things is the Gregor Research Consortium. This is an NHGRI funded consortium, and it's an abbreviation for genomic research to elucidate the genetics of rare disease. There are a number of individual institutes that are part of this effort, and as you can see, they all have their different types of participant recruitment, their different ways of doing sample prep, the experiments that they are running, and the data that they are producing. In the end, though, everything is modeled according to the Gregor data model. And together, there is now 2 releases. They are approaching their third release, and there are several thousand genomes, whole exomes, long reads, short reads. There's a ton of data here. So how does one go into the Anvil and actually make use of all of this? The Gregor Consortia have put together a demonstration notebook that reviews how to identify samples in a joint call set by using the Anvil Bioconductor package and dplyr. So what this means is you can bring these data sets into dplyr and start manipulating them the way that you know and feel comfortable doing. And so Bioconductor is really important to making sure that a lot of these activities are moving forward with strength. Galaxy is really working very hard at making sure some really sophisticated workflows are also available in the Anvil platform via Galaxy. And of course, there's a lot of opportunity to model out the costs of what some of these applications that individuals want to run and what they might cost. So then there's opportunity to start looking into very specific compute infrastructures for your tool, for your job, to make sure that you're getting the best results and the most with the least amount of investment. And of course, the team is now looking to tap into GCP batch to allow for optimized workflow execution. So these things are on the go and in development. Some other things happening in the Anvil world right now is the NHGRI R25 award for the Monet workshop. This is a multi-omics network analysis workshop. So this was actually held on the 8th and through the 14th of this month. So just a couple days ago. And a number of workshop attendees traveled to UC Anschutz in Colorado to attend this workshop. And I think that there's funding to bring students into town to attend this workshop. If anybody is interested in being part of this, there is another Monet workshop coming up next year in 2026 if you want to learn how to use the Anvil platform. These folks, the attendees here, commented that the Anvil was really interesting to them for the secure platform, the data access, the ability to bring your own data. So you don't have to analyze just the data there. You can combine with your own data sets and analyze that there. Everything is protected and by default private. So you can work in your own workspace until you are ready and then make it public and share it with the world. It allows, people were commenting on the flexibility of tools, the quality of the community, and the number of resources that were put together to help get started. Another effort that I'd like to point out is Grads4C. This is Genomics Research and Data Science Center for Computation and Cloud Computing. And Grads4C is a national education hub empowering researchers and students and trying to teach the next generation of students how to be cloud ready, how to analyze genomic data science in the cloud, a cloud-first approach. So there's an educational hub and training network set up so that there are some funded institutions that will come and collaborate with this hub and then bring these educational materials to their own institutions and help spread the word of cloud-based genomics computing. There's also a number of collaborative research and mentorship roles that have been generated. I think that there are some fellowships. So what about you? What if you want to use Anvil? Anvil's on GCP, so that means you've got to pay for what you're going to compute. Where do you get the money to do this? I want to point out an NHGRI funding opportunity announcement, and this is called the Earned for Funding Analysis. This is called Enhancing Reuse of NHGRI Data Assets, or Earned. And we are already going through NHGRI is already going through the first round of this with submissions that were due earlier this year in February, but there is another round of applications coming up in February of 2026. If you are interested in getting some cloud credits to continue your work, this is a great idea. Now, how can you learn more about Anvil in practice? Tomorrow, on the 25th, from 2 to 3 p.m., Fred Tan will be leading a workshop, Anvil Secure Scalable Computing for Controlled Access Data. Natalie will be giving a talk at the session 4, called Training and Education in the Cloud with NHGRI Anvil. And then, of course, there's tons of resources for you at anvilproject.org. There's monthly demos where where researchers come and show you how the platform is used, and then you can come and ask questions. And there's tons of videos and demonstrations on Anvil on YouTube as well. Also, finally, I want to highlight that the first Anvil Community Conference was hosted here in Cold Spring Harbor, and we're really excited to have the second one coming up in September 4th and 5th. This will be at Vanderbilt University Medical Center. Also, Anvil team will be at ASHG and Genome Informatics later this year, also here at Cold Spring Harbor. So thank you for your time.